Today's session, talk a little bit about stats and then just some background in terms of definitions, what different terms mean, which is really fundamental knowledge, but it's really helpful and it often gets skipped over in most stats courses. And so it leads to people making kind of poor decisions down the road when they use statistics. And then hopefully we'll have enough time to cover some of the basic descriptive statistics and just give you some ideas as to things to be aware of. Okay, so why use statistics? Why do we bother with this stuff? Nobody likes to take it in school. I taught it for 13 years, and everybody hated taking my class. But if we're going to do anything with data, we have to be able to make sense of that data. And so statistics are really just a set of tools, and there's lots of them, and we're going to cover just some basic ones over the couple of sessions. But they're just a set of tools to help us make sense of data because data tends to be variable and it's not clear cut. Even in well-designed experiments, we have other things going on. And so we have to make sense of the numbers. So whenever we do an experiment or gather data, we're trying to find out an answer to a question. And so it's always important to remember what the question was and what we were trying to answer. And then we gather data. And the statistics lets us go from that mass of data, just the raw numbers, to going back to the question we're trying to ask and what evidence do we have then to answer that question. Okay, so that's why we use statistics. That's why we can't just report numbers and say now we know what the answer is. So we're going to go over a couple of background pieces now in terms of how we get data because it's really important to understand that part when you start calculating statistics. And so the first thing we're going to talk about is kind of what sorts of studies you might do to gather your data. And they fall on a continuum from two extremes. One is, yeah, I can kind of highlight things with my mouse. One is experimental data, and that for some people is the gold standard. That's a lot of what we try and do in the CAWT, and that's where the researcher has complete control over treatments and what happens to either your individuals if it's a human-based study or your samples if it's a sewage-based study or whatever it might be. So an experimental study is kind of considered the gold standard because you can control for a lot of variability. At the other end of the extreme is observational work where you just gather data and you don't necessarily have much control over what happens to any of the units in the study. Right? So if we think about COVID, to try and put this in context, all the public health data we keep getting and watching, that's observational data. Nobody's controlling who gets infected. It's just watching to see what happens in the population. All the clinical trials to see if a particular vaccine or a particular treatment has an effect on COVID, that would be an experimental study, or should be, as long as nobody's interfering. It should be an experimental study where we have control over what the patients are like. We make sure all the patients are as similar as possible, or we account for those variations from patient to patient. Now, this kind of two ends of an extreme. So we can have data that comes from a study that is mostly observational, but we have a little bit of control, right? So we might pick and choose which, say, lakes to get data out of, but then we don't have much control over what's going on in the particular lake. So it's somewhere in between an experimental and an observational study. Important to know how your data is being generated because this will impact how you draw conclusions from your statistics. When we start to get data, we need to think about how we're getting a hold of the data. And so we always gather data from some sort of population, and it isn't necessarily a biological or human population. It's just a collection of entities. And so it might be pipes in a system, in a municipal system. It might be sewage samples from an output of a wastewater treatment plant. It might be sediment samples from a lake in the Yukon, just to kind of think of some random examples that popped into my head. And so what you want to do is kind of figure out what is the thing I'm getting my data on. So what's the entity and what's the limit to that data? Is it sediment data that I want to say something about a particular chain of lakes in the Yukon, or is it sediment data and I want to say something about all lakes in northern Canada? And that's going to impact how you try and get your data, right? And so you have to think about what is the limits to my data, because that then limits what you can say in your conclusions. Okay, and so once we figure out what population we're getting data from, then we have to figure out how we're going to get the data. And this is a very overlooked part of statistics, how you get your samples. And ideally, what you'd want to do is take a subset of the data. And if you had a really small population, and the example I always use in class was if you wanted to know something about condors, you could possibly measure it on every single condor because there's only a handful of them left in the wild. But for most populations, you can't measure every single individual, so you need to take a sample. But how you take that sample can influence the accuracy of the data. And so ideally, you'd want to take a representative sample, and that usually involves some sort of random sample. Okay, so you want to be able to choose your individuals at random. And by individuals, I mean whatever unit it is you're doing your testing on, whether it's people or wastewater samples or pipes or what it might be. And this is actually a pretty tricky thing to do, and most people don't do a very good job. And so a true random sample would mean that all individuals in the entire population are equally likely to get sampled. So you would have to have some way to actually randomly pull individuals out of the population. And so if you think about, say, sampling lakes, say you want to do some sort of lake water chemistry, to actually randomly choose lakes, you would have to identify all the possible lakes, make a list, randomly choose a number from that list, and then that's one of the lakes you'd go sample. And there was a study way back in the 80s that looked at all the data that was gathered by limnologists to study lakes. And most of the lakes that were ever sampled were within a 3-hour drive of a major research university in the States that had a boat ramp. So many, many lakes were never sampled because they were hard to get to. So it wasn't a true random sample. So often what we do is we use a haphazard way of sampling where we try not to have any pattern to how we sample and assume that it's actually random. And one of the illustrations that I would always often use to say, if it's not a random sample, you might not get a good idea. It's if you imagined you were on a university campus and you were trying to sample average height of students for some reason. Well, if you took your sample of all the students leaving the gym when basketball practice got out, you wouldn't get a very good representative sample. So how you draw that sample can really skew the data. And so and the data underlies all the statistics. There's no statistics you can do on data once you have it to fix problems with your sampling. So if your sample is biased, all your statistics will be biased. There are some things to think about when you're doing your random sampling. So if you know something about the system, you might stratify your sample. You might sample big lakes and little lakes to make sure you've got a good mix across there. So there are ways to deal with that in terms of stratified sampling, different ways you can do random sampling. I won't go much more into detail there. There's lots of details that you could think about. But the important point is to understand how you got your data and was it a random sample or not. Okay, so now we'll get to some definitions. We've been talking about some ideas like data, and data is simply some sort of numerical fact that you can gather about an entity in your population. So it might be height, it might be weight, it might be BOD, biological oxygen demand, it might be length of a fish, it might be biomass, whatever it might be that you could measure. Once you have that data, you can measure something from that data. So that would be a variable. And a variable is anything you can measure that changes from one observation to the next, from one datum to the next. So it might be pipe thickness, it might be biological oxygen demand, it might be height, it might be length of a fish, or whatever it might be. With those variables, then we can start to try and describe them, and that's where we can start to get answers to our questions. And as we describe them, we can think of a variable having both a parameter, which is the true value of that variable in the population. And we never know the parameter, and then we try to describe that parameter with a statistic. So for example, we might think of, say, a fish sample, and we want to know what is the weight of the fish. And so for each for that population, which might be fish in a tank with a certain feed, for that population, we can ask how much does each fish weigh? And there's probably a true value of how much each fish weighs, and that would be the parameter, and we might describe that as the average weight of a fish. We can't actually know that true parameter, and so we measure it and estimate it with a statistic, and that would be our sample average. So, okay, so I mentioned variables. It's important to understand the different sorts of variables because those will dictate what sort of statistics you can calculate. And so there's two main categories of variables. There's categorical variables and numerical variables. We'll start with categorical. Categorical are variables that describe membership in a group, and they don't have a size. And they can come in two sorts: ordinal scale, which are categories that have an order, and you can put them in order from the smallest category to the biggest, and nominal scale, where they have no order and cannot be ranked. The other sorts of variables, and these are the variables that we tend to see more of, are numerical variables. These are variables that have measurement, they're quantitative, and they have a magnitude. So there's little values and big values. And there's a couple of different things to think about with this. One is that you can have continuous data, so that can take on any value. The other would be discrete data, and that you can only get certain numbers, right? So kind of like for those of you that took math courses, real numbers versus integer numbers. The one other thing we need to think about with numerical data is what scale was it measured on? And it's important because it can have a little impact on what you can do with the numbers. We have three scales that we have to keep in mind. One's a ratio scale. That's a constant interval size and a true 0 point. What that means is that if you measure a variable as 0, it really is a 0. And so that would be something like height. So if a plant was 0 centimeters tall, it really has no height. And each centimeter that you go up in height is the same interval. So that would be measured on a ratio scale. Most of our numerical variables are measured on a ratio scale. An interval scale is very similar to ratio, except that the 0 isn't a true 0. So it's an arbitrary 0. Can anybody think? Right? The more tricky one is things that are measured on a circular scale. And a circular scale refers to a scale that wraps around on itself. And so something like time of day, right? So midnight becomes midnight again after 24 hours. And so that's a circular scale. But just as you start thinking about data, you need to decide, is it categorical or numerical? And if it's categorical, is it ordinal or nominal? And if it's numerical, is it continuous or discrete? It's important to recognize those things because they'll dictate what you can do with that data, both in terms of describing it, and then once we get to tests a little down the road, what tests you can use on the data. And I've seen students try and do tests on data that you can't do it on, and they get wonky answers, and they try and make sense of it. So it's really important to recognize what sort of data you have. Okay, thanks everybody. I've got another thing happening at 11, so I'm off to my next meeting. Have a good day, everyone.
Thanks, Brett.
Thank you.
Thanks.
Bye.